Meiosis is a unique type of cellular division for gametes, or sex cells. On this first slide, we see the gamete in the stage called interphase. During this stage, the cell grows and synthesizes, or duplicates, DNA. This is the longest stage of the cell cycle where the gamete spends most of its time. Toward the end of interphase, the gamete prepares itself for meiosis. This slide shows the overall process of meiosis that gametes undergo. Meiosis starts with the mother cell, as shown in the previous slide, and ends with the production of 4 daughter haploid cells, which are genetically different than the mother cell. Meiosis is split into 2 phases. Meiosis 1 is shown here. In this phase, the mother cell splits in half and undergoes cytokinesis, creating 2 daughter haploid cells. The stages include prophase 1, metaphase 1, anaphase 1, and telophase 1. In the second phase of meiosis, the 2 daughter cells are both split in half, leaving 4 daughter cells, each with 1 chromosome each. The stages include prophase 2, metaphase 2, anaphase 2, and telophase 2. In prophase 1, the first stage of meiosis, chromosomes come along one another. The nuclear envelope begins to deteriorate and crossing over occurs between the sets of chromosomes. In metaphase 1, crossing over is shown as complete. The tetrads of the chromosomes align along the central equator of the cell. Centrioles move to opposite poles and the spindle fibers begin to form. During anaphase 1, the cell starts to be pulled apart by the centrioles and spindle fibers. Unlike mitosis, the centromeres of the chromosomes remain intact. Homologous chromosomes do, however, move to opposite sides of the cell. In telophase 1, the nuclear envelope begins to reform around the chromosomes. The spindle retracts and the 2 daughter cells are now haploids. The cells enter an interphase-like stage called interkinesis, where the cleavage furrow is eventually separated during cytokinesis. DNA does not re-replicate during this phase. Now that the first phase of meiosis has been completed, the cell moves on to meiosis 2. The first stage is prophase 2. This stage is very similar to prophase 1, except it is with 2 daughter cells. The nuclear envelope begins to deteriorate, and the spindle begins to attract one another from the centrioles. However, crossing over does not reoccur. Metaphase 2 is much like metaphase 1. The chromosomes line up along the central equator of the cell, and the spindle fibers form around it at opposite poles. During anaphase 2, the chromosomes split in half and are pulled to opposite sides of the cell. In the last stage of meiosis 2, telophase 2 occurs. In this stage, you can see that there are 4 daughter haploid cells that have been created. The nuclear envelope begins to reform and the spindle fibers retract. The end result of meiosis is 4 daughter haploid cells, each with their own unique set of DNA, different from the original mother cell, thus creating genetic variability. With humans, each gamete created only has 23 chromosomes. Therefore, when the male and female gametes interact, a resolved 46 chromosomal number will occur.